Chapter 4, Nucleic Acids and an RNA World. Nucleic acids will be the second type of macromolecule that we discuss. Remember, in the previous chapter we talked about proteins, so the second important macromolecule are nucleic acids. Nucleic acids store the information that encode life. So we need to start by asking what is a nucleic acid, and then compare and contrast the two types, DNA and RNA. We'll talk about their function in the cell, and lastly we'll talk about the evolution of these macromolecules. Was it a DNA world first or an RNA world? Chemical evolution led to the production of molecules that could self-replicate, and this is the tipping point for life. So we'll go through this chapter and we'll take a look at both information molecules, DNA and RNA. DNA stands for deoxyribonucleic acid, and DNA stores genetic information and is replicated with the help of proteins. But the idea is that it was an RNA world first before it was DNA, and this is proposed because RNA is both stored as a genetic information, but it can also catalyze its own reaction. So many scientists believe that RNA was the first self-replicating molecule. It's often called a naked replicator. Okay, so what is a nucleic acid? A nucleic acid is a polymer. So remember what a polymer is? A polymer, poly means many, so it's built of many monomers, and a monomer is the building block. So in this case, for nucleic acids, the monomer or nucleic acid, the monomer for nucleic acids is a nucleotide, and this nucleotide has three components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. The phosphate group and nitrogenous bases are bonded to the sugar molecule. So we can talk about the two types of nucleotides, ribonucleotides and deoxyribonucleotides. But I think in reality, when people are talking to each other, we just say nucleotides, and we make reference to either RNA or DNA. But what are some of the major differences between the two? Ribonucleotides have ribose as their 5-carbon sugar, and they also have a hydroxyl group or an OH group bonded to the 2' carbon. In just a second, we're going to see a diagram, and I'll show you what the 2' carbon stands for. On the other hand, DNA, deoxyribonucleotides, has deoxyribose as its 5-carbon sugar, and deoxy means lacking oxygen. So it has a hydrogen instead of an OH group at the 2' carbon. Both sugars have OH groups bonded to the 3' carbon. There are 4 nitrogenous bases, and they fall into 2 basic groups, the purines and the pyrimidines. So the purines have 2 rings, and the nitrogenous bases in this group are adenine and guanine. A mnemonic device to remember that is pure as gold. As gold reminds us of adenine and guanine. The pyrimidines only have 1 ring, and the nitrogenous bases in this group are cytosine, uracil, thymine, and we can think of this as a mnemonic device: cut the pie, cut the pie. This diagram clearly states almost everything that you need to know about the building block, the nucleotides for both DNA and RNA. So let's look at A, the first one on the left-hand corner. Notice that a nucleotide has 3 components: a phosphate group, a 5-carbon sugar, and a nitrogenous base. And there's 4 nitrogenous bases. Now let's look more closely at the sugar. Notice that at the corners in a clockwise fashion, that each of the corners have 1', 2', 3', 4', and then 5'. Each of those corners of the ring represent a carbon. So 1' is the first carbon, 2' is the second, third and fourth and fifth, and so on. So each of these carbons are labeled, and this is how we will know the directionality of our huge macromolecule. Notice in the second square, we have sugars in block B. And notice we can see the difference between the sugars in DNA and RNA. So ribose is found in RNA, and notice that at the 2' carbon, the second carbon, that there is a hydroxyl group, OH. But if you compare it to deoxyribose, notice that there is just a hydrogen that is highlighted, connected or bonded to the second carbon in the ring. Lastly, if you look on the right-hand side, you'll see the different types of nitrogenous bases. Keep in mind that there are 4. Apple tree chewing gum, that's for DNA, A, T, C, and G, and then A, U, C, G for RNA. So notice how they're grouped into pyrimidines and purines. Cytosine, uracil, and thymine, these are actually pyrimidines. If you look at purines, it's adenine and guanine, and notice they have double rings instead of single rings. Could chemical evolution result in the production of nucleotides? The idea that amino acids could have been synthesized in early Earth is accepted by biologists, but what about the building blocks of nucleic acids? Simulations show that nitrogenous bases and sugars can in fact be synthesized. So recent work has focused on deep sea thermal vent reactive minerals, and from there we see that ribose can bind to reactive minerals down in deep sea vents. How do nucleic acids polymerize to form nucleic acids? Well, it's the same thing. They use condensation reactions. And you'll have to know this bond. It is a phosphodiester linkage. So the phosphodiester linkage links the phosphate group on the 5' carbon of one nucleotide to the hydroxyl group in the 3' carbon of another. And from there we get a connection or a bond between two nucleotides. So we can form RNA and we can form DNA. If we look at this diagram, we can see that there is a condensation reaction occurring in the very middle. Let's see if I can highlight this. So here's a condensation reaction. OH and H will give us water. When water is released, a bond is formed, and this bond is called a phosphodiester bond. So the bond is formed between a hydroxyl group and the phosphate group. So this is what links one nucleotide to another. So phosphodiester linkages form a sugar phosphate backbone, and notice that there is directionality. This goes into the 5' to 3' direction. So let's recall where we get 5' and 3'. These numbers come from the numbering of the carbons found in ribose and deoxyribose. It's found in the 5' carbon sugar that is found in nucleotides. So it's saying that one end of a DNA or RNA strand has a 5' phosphate group. The other end has an unlinked 3' hydroxyl group. And this is really important, and you're going to need to know this for later on in the semester. So here's an example of a primary structure of DNA. Keep in mind that when we refer to the primary structure, we're looking at the sequence of nitrogenous bases. And this example shows directionality. 5' and then it has ATTAGC 3', showing that one side has the phosphate group, the 5' end, and the 3' end has the OH or hydroxyl group. This drawing reinforces this idea of directionality. Notice at the top of the diagram that we have a phosphate group, and it is connected to the 5' sugar in the 5 carbon sugar. So we have 5' at one end, and at the other end we also end with a sugar, and that sugar is that 3 carbon 3' carbon has the hydroxyl group. So notice the directionality of nucleic acids. So nucleic acid polymerization can take place in cells by using enzymes. So enzymes have to be used to catalyze the reaction to actually build a DNA molecule. So where does the energy come from? Because keep in mind that when you're building, going from a small molecule to a large molecule, you need a source of energy. And in this particular case, the energy comes from the monomers themselves, from the building blocks. So they're not really nucleotides, they're nucleosides. And the nucleosides means that each of the nucleotides has a triphosphate tail. In other words, it is activated. So we can say that a nucleoside is an activated nucleotide. The most famous activated nucleotide of all time is ATP, adenosine triphosphate. So that triphosphate tail gives energy to the building block. So because of this, energy is released when activated nucleotides polymerize, making the reaction a spontaneous one when DNA or RNA are being built. In this figure 4.4, you can see an activated nucleotide. It is ATP. What makes it activated is the triphosphate tail. So why does the triphosphate tail have so much energy? If you take a closer look at the triphosphate tail, you'll notice that there's 4 oxygens and they all have a negative charge. But you know from chapter 2 that negative charges repulse each other. So that must mean that those bonds holding each of those phosphate groups together must be particularly strong. So when any of those bonds are broken, a lot of energy is released, more than normal, because they're extra strong bonds holding those negative charges. And down below you can see that the activated nucleotide or nucleoside is broken or hydrolyzed in the presence of water. The 2 phosphates break off and one is left as the nucleotide that is going to be used to polymerize and form either DNA or an RNA strand. What is the nature of DNA secondary structure? Early data provided clues to DNA's secondary structure. So back in the 1950s, when they were trying to figure out exactly what the DNA molecule looked like, they had several clues. So early scientists found that the number of T's always equaled the number of A's, and the number of C's always equaled the number of G's. This is called Chargaff's rule. And so this sort of gave a clue as to what it might look like. Then later on, Dr. Franklin did X-ray crystallography, and it sort of looked like looking at the end of a Twizzler. If you look at the end of the Twizzler, you see that it's spirally, so this gave a clue to the double helix. So James Watson and Francis Crick determined that 2 strands are held together by hydrogen bonds, between pyrimidines and purines. And they know this through complementary base pairing. And that's why the number of A's always equal to the number of T's and C's always equal G's. It's because of complementary base pairing. They also found that DNA strands are anti-parallel. One strand goes from the 5 to 3' direction, and the other strand goes in the opposite direction, 3 to 5'. Anti-parallel strands predicted to twist together to form the double helix. The sugar and the phosphate backbone faces the outside, and the nitrogenous bases face the inside, and they look like steps to a ladder. In this figure 4.5, you can see why it's complementary base pairing. Because if you had 2 purines together, 2 double rings, then they would overlap each other. If you had 2 pyrimidines or single rings together, they wouldn't meet in the middle. So what's just right is having 1 pyrimidine and 1 purine. In B, we notice that actually the nitrogenous bases in the middle are held together by hydrogen bonds. For the test, you don't have to know whether it's 3 hydrogen bonds or 2, but you do have to know that the double strands are held together by hydrogen bonds. And in the last frame, in part C, we actually see the anti-parallel strands. Notice one strand is going from 5' to 3', and the other strand is going in the opposite direction. And notice here we can also tell by which way the 5-carbon sugar is pointing. Notice on one side, the left-hand side, it's pointing up, but on the right-hand side, it is pointing down. DNA is put together like a ladder. As I mentioned before, the sugar phosphate backbone faces the outside. It's like the side rails of a ladder. Bases face inward, and they're like the steps to a ladder rung. Hydrophobic interactions cause double-stranded DNA to twist into a helix, and van der Waals interactions stabilize strands. Remember, we've talked about van der Waals when we were talking about proteins. These are mild interactions between hydrophobic molecules. DNA also has 2 size grooves, a major and a minor groove. In this drawing here, you can see the major and the minor groove. Here we can see a major groove, and here is a smaller one, which is a minor groove. So as it's twisting, the twists aren't exactly equal to each other. There's major and minor grooves. Here are some diagrams of what the DNA molecule looks like. The tertiary structure of DNA. Later on in this semester, we'll be talking about cell reproduction, mitosis and meiosis. And right before mitosis and meiosis, there's a compaction of the DNA molecule. How does it become condensed? Well, it supercoils, and that is its tertiary structure. It supercoils around a protein, and these proteins are called histones. So DNA wraps around DNA binding proteins called histones to supercoil, and this is often found right before mitosis and meiosis. Watson and Crick's model also revealed as DNA as a biological reservoir for information. So the major function of DNA is to store information. In other words, it has the information that makes you you, or a cockroach, or a goldfish a goldfish. And we already know that the building block of DNA is the string of nucleotides. And these nucleotides have one of 4 nitrogenous bases, A, T, C, or G. So this order is very important. It's like the order of letters in a word. It has meaning, and then a string of words makes a sentence, and that too has specific meaning for us. So that would be a great analogy of looking at the sequence of bases, and that is coding for a specific trait. DNA replication has 3 steps. First of all, the 2 strands have to separate. So the 2 strands have to unwind, and then the hydrogen bonds need to be broken. After that, we can say that one strand can be used as the template strand, and the other one is the complementary strand. But because of complementary base pairing, if we know the nitrogenous bases on one side, we have to know the nitrogenous bases on the other, because A always complements T and C always complements G. In this example here, we see the 3 steps. Notice in the first step, DNA is unwinding and the hydrogen bonds are being broken. Then one strand becomes the template strand and the other one is a complementary strand. So the old strands become actually the template strand, and we can see new strands being built because of complementary base pairing. In the last frame, you see that there is the two new strands of DNA are composed of one new strand, one old strand, and one new strand and one old strand. So this is due to complementary base pairing. The DNA double helix is also a very stable structure. So it is highly stable because it has a double helix. So these linkages actually protect the nitrogenous bases that are inside. And it says functional groups participate in chemical reactions, but it makes the molecule stable and resistant to degradation. And isn't that what we want out of our information molecule? We want it to be stable, in other words, not very reactive, and we definitely don't want it to degrade. But because of this stability, chemical stability, is it possible that DNA could actually be that tipping point that caused that caused replication in information for the first cell? So that's a big question. So because of its stability, maybe RNA is maybe the first molecule that we need to look at. So the primary structure of RNA is the sequence of the different types of nitrogenous bases. And remember, instead of apple tree chewing gum, it's AUCG. And so how is it that RNA differs from DNA? First of all, instead of having deoxyribose, it has ribose. Number two, it has uracil instead of thymine. And its 2' hydroxyl group on ribose is more reactive than the hydrogen. So for step 3, we can say RNA is much more reactive, chemically reactive, than DNA, and it's a lot less stable, step 4. RNA's secondary structure results from complementary base pairing. So how can that happen when RNA is a single-stranded molecule? Well, it can fold on itself. And when it folds on itself, it is called a hairpin structure. When it folds on itself, actually we have complementary base pairing. So A always pairs with U, and C always pairs with G. And when this hairpin structure forms, like a bobby pin found in a person's hair, we can see that the strands are anti-parallel, similar to what you would see in DNA. And here's an example of a hairpin structure. Notice when the arms of RNA have folded on each other, we have complementary base pairing. There's also a tertiary structure to RNA. So it can continue to fold on itself, and when it does, it can form diversity in size and shape and reactivity. So there is a tertiary structure to RNA. One of the most classic tertiary structures is a molecule called transfer RNA. So there's messenger RNA, ribosomal RNA, and transfer RNA. And transfer RNA, we're going to find out later on in the semester that transfer RNA actually carries amino acids to the ribosomes to build proteins. So we'll be talking about this later, but this is because of this folding of RNA, as you can see this in this diagram. So here's an example of a tertiary structure, and this is a tetrahymena ribozyme. So we'll talk about what a ribozyme is in just a moment. But before we do, I love this summary table 4.1. If you look at this table, it compares DNA and RNA, and it looks at its primary, secondary, and tertiary structure. It gives a description and then shows you a diagram, so you can't go wrong by looking at this. If you understand this summary table, you've understood my discussion up until this point. RNA is highly versatile, and we know this because it can form a lot of three-dimensional shapes because of its tertiary structure. Not only that, it's capable of catalyzing reactions just like proteins. So this is where it gets its name, ribozyme. My best analogy, believe it or not, is the movie RoboCop. RoboCop was a human cop and he's part robot, so he's RoboCop. So RNA is very versatile. Not only does it carry information, but it can catalyze or speed up reactions like a protein. So it's called a ribozyme. It's like a ribozyme is like RNA and a protein is a ribozyme. So here we are, ribozymes. They're RNAs that can catalyze reactions. And keep in mind, when we say catalyze reaction, is they help speed up a reaction, but they don't get involved. So it has active sites just like proteins and specific proteins like enzymes. That's where ribozyme comes from. And so it has the ability to catalyze phosphodiester bonds, giving rise to the possibility that RNA can actually replicate itself. So what is this idea? So for life to begin, remember there's a chemical evolution before a biological. Remember, when we talk about a chemical evolution, we're talking about moving from very small molecules to these huge macromolecules. And now we've talked about two of them. The first one were proteins, and the second one is this chapter itself. It's the information molecules, nucleic acids. So here we have the tipping point for the first cell is to have a naked self-replicator, a molecule that can actually replicate itself and have information, and this molecule is RNA itself. So you need to have a template, and you need to have the ability to catalyze the reaction, and that's why many RNAs are called ribozymes. So that's why many researchers believe that it was an RNA world first before it was a DNA world. In this example here, we see that RNA can actually replicate itself. So how is this done? You see that RNA is a single strand, and that single strand acts as a template, and it builds together this secondary strand, as you can see in gray. So this gray strand is now the copied strand, and this copied strand is now going to act as the new template to actually build more RNA. So it isn't just a theory. They've done several experiments, and these experiments show that through natural selection, or should we say artificial selection in the lab, we can get RNA to actually improve its replication ability, to improve its ability as a ribozyme. So in one study, they generated RNA molecules that could actually catalyze an RNA reaction. And this one was with replicase, a type of RNA. So what they did is they looked at 14 nucleotides, and they could actually get RNA to replicate itself. In another study with just uracil bases, they got RNAs to actually replicate the string of uracil bases. So this showed that actually RNA can carry information and it can catalyze its own reaction. So this is awesome findings. In an RNA world, this is what we believe sparked the evolution of life. So now this is the tipping point of having a naked self-replicator. So all that we're missing is a membrane. So once a membrane encapsulates this naked replicator, we almost have the first cell. And this is important. So most modern ribozymes do aid in protein production, and if they were removed, then RNA probably, a lot of proteins wouldn't be made. So RNA probably preceded proteins. So RNA is a very ancient macromolecule. So keep in mind that RNA has three characteristics of life solidly in place: information processing, replication of heredity information, and evolution by random changes in nucleic acids. And this is the end of this chapter.